So yeah, so what I've got here is, I guess, really a collection of thoughts and reflections around the current state of play of many medicinal chemistry departments, particularly with the focus of the interactions between computational chemists and computational tools with medicinal chemists. And then some, pictures of what a, shorter term future might look like, following with some thoughts of what a longer term future could look like too. So yeah, you heard a little bit about my background. I thought I would go over that again. I think it's kind of relevant here because, firstly at the top there, my training as an undergraduate was really as a synthetic organic chemist. And, I loved synthetic organic chemistry. And this is the same background that many medicinal chemists have. Yet following that kind of initial training in synthetic organic chemistry, I really took a change of direction to become a computational chemist. Continue to kind of maintain a lot of that synthetic knowledge through problem sessions and things like that, but very much a paper synthetic organic chemistry. But then, did a PhD in the application of computational chemistry to protein-protein interaction targets. And this was kind of like running mini drug discovery projects where, I was selecting molecules through virtual screening, and then we were doing kind of early med chem on those. So we were finding derivatives that were commercially available, designing compounds, getting things synthesized by often students in that case. And so, went on that journey from synthetic organic chemist to kind of computational medicinal chemist, but, with some medicinal chemistry skills, but not as much as I would say I've got now. And following that, joining Lilly, some of the key skills I picked up there was from working on projects and seeing that the needs of those projects rarely were in optimizing potency, which was kind of the background I'd had, a lot of structure-based design, and realizing that the control of properties and particularly ADME and PK was absolutely key. And so, gain skills in ADME, and I think they, I see those as key skills of a medicinal chemist. And, working on projects, supporting those and applying those skills and learning at the same time. And then Lilly shut their site in the UK, and then I joined UCB for the sort of last 16, 17 months, where kind of continuing in that mold, working on projects. And so I kind of really see myself as a medicinal chemist, although formally I'm a computational chemist. And so I kind of sit between those disciplines. And so what I've got here is the kind of reflections on that experience and seeing how more traditionally trained medicinal chemists or more traditionally trained computational chemists, how they've often approached things, and then what the future might look like, where these disciplines come closer together, where I've seen some of the benefits of doing them. So in terms of what I'll talk about, so firstly, I realize that not everyone here will necessarily have a drug discovery background. So there's a very brief intro into kind of drug discovery and where some of the pieces I'll talk about fit in. There'll be this historical and evolving perspective on both medicinal and computational chemists. And then given that kind of historical setup and what often persists today, what does the next generation of kind of computational medicinal chemists look like, and what do computational tools look like in that space, and with a particular focus on how they might interact with users, with a particular focus on automation in that space. So before we start, actually a disclaimer. So, these are my own personal views based on, the background I described, and they're not, a UCB sanitized view of the world. And they, may or may not agree with the wider UCB opinion. So in terms of the, high level, what happens within drug discovery, many drug discovery projects, not necessarily all, is that within medicinal chemistry I've kind of got highlighted in the yellow box, where we within medicinal chemistry work with obviously our cross-functional partners. But we would often inherit a target that's been selected based on a biological hypothesis that if we manipulate this target in a particular way, that molecule may then impact human disease. And our job within medicinal chemistry is to deliver a molecule that can act via that hypothesis that can go into humans to test it. And so what we would do is go through hit identification, try and find something that has an aspect of what we're looking for. And then it's an optimization challenge where we go into hit to lead, where we're trying to take those first hints of activity and move to something that looks like it has the legs to deliver that ultimate candidate molecule to go into patients. As we refine these molecules down, the space narrows, and I'll talk about some of that in a second. But this next stage is kind of lead optimization, where we're really trying to narrow down on that chemical space into getting closer and closer to our candidate molecule until ultimately we deliver that candidate. And we would, select that and go through all the various, hurdles and necessary processes to then take those molecules into the clinic and actually test the original hypothesis that may have been generated many years previously and see whether it holds up. In terms of what a candidate small molecule might look like, this is often the situation we find ourselves, not necessarily always. And so what we have on this plot, we have time on the x-axis, where time 0 is the time we would take a pill. And then we've got the concentration of that molecule that we've taken, the unbound drug concentration. So when you take any medicine, it will bind to particularly plasma proteins. And so we think about the unbound concentration here that's available to engage with our target. And so when we take a pill, there's a period of time where that molecule is absorbed. We take it, say it's taken orally, travels into the gut and it, absorbs through the intestine and, has a little route through the liver and then gets into the sort of systemic circulation. And so ideally, we're trying to find a molecule that is absorbed and then has a continuous presence for the duration of time that we're looking for, and that concentration is above what we think we need to have efficacy. So this minimum concentration here, this is like the activity that we think we need, the activity on the target, and so we need to achieve a concentration higher than that for the duration of time we think we need this. If this was, say, a pain project, and this was like migraine, you feel a migraine coming on, you might want to take a pill and have this engage for 2 hours, and then you'd stop it, and hopefully your migraine might have gone away. If this was a chronic disease where we need to cover the target 24 hours a day, this time period could be 24 hours. And in that sense, we might take a pill and then we would take another one, and you get into, challenges and complexities of multiple dosing. But, this is a simplification here. So if a concentration we want to hit that's related to the activity on our target, the concentration of that molecule in vivo, and then there'll be a high bar of where if we reach a certain concentration or a certain concentration for a period of time, then we might see some toxicity associated with the, engaging whatever target that might be. And that could be a target anywhere in the human, basically. So it could be that you're targeting a molecule in the brain, but you're causing toxicity in the liver, whatever that might be. And within medicinal chemistry, these are the handles we have to turn. We design molecules to try and move all of these lines so that you get to something that vaguely resembles this. When we think about potency, we're thinking about moving this green line up and down. But then we also have the ability to change the pK of our molecule. We can think about the Cmax, we can think about the half-life, which will relate to the amount of time where, how flat this line is. The flatter it is, the longer we're likely to engage a target for. Or if our concentration gets too high, we start to hit tox, and this tox could be related to a direct target that we know about, or it could be related to non-specific binding. If your molecule is, say, quite lipophilic, maybe if it's basic, it might be hitting targets we don't want it to. And the thing is, when we make a change on our molecule, all of these things change at the same time. So that's why one of the reasons why the problem is quite challenging. And we're thinking about how humans think about multi-parameter challenges like this versus the role computers have in that same scenario. That's why I think it's a kind of an interesting thing to talk about. And so if we think about drug discovery from, using terms that are a bit more related to, computer science, drug discovery can really think about as a sampling problem, where at the beginning, when we're thinking about trying to find a hit, we're really thinking about the entirety of what we might call chemical space, every molecule you could possibly imagine. And the thing is, this space is absolutely vast. It's well beyond what a human can even concept in terms of how big that number is. Yet the molecules we're trying to find ultimately as a candidate, might be a single molecule, it might be a small number of related molecules. And so at each stage, we're narrowing down the size of the space we're considering to go from, in hit ID, we have every molecule we could possibly consider. And you've seen various numbers of the estimate of this, anywhere from the sorts of numbers I have here, around 10 to the 30, through to numbers on the order of 10 to the 60. That's a ridiculous number of molecules. And then as we find something that shows a hint of what we're looking for, we're narrowing the space. So we have some hits, we have a few scaffolds that we're interested in. And then the space around those scaffolds, we could take those in many different directions. But that space is still very vast. Once you start thinking about the different groups you could have at multiple positions, combinatorial explosion kind of happens, and these numbers get very big very quickly. As we narrow this down, we begin to understand, well, what spaces are tolerated by our target, and what spaces are tolerated from an in vivo perspective in terms of that PK, in terms of the potency line at the bottom on the plot we had and the shape of the curve. We might have one, we might have two scaffolds at this point, and the space contracts, but it's still a decent size. I would say it's at least 10 to the 5, so 100,000 molecules. It's probably more like many million, if not tens of million, because of the size of chemical space and the things we could consider. And then we're trying to filter this down to ultimately a candidate. And so, if I think about sampling, think about what tools are good at sampling. What would I use in a sampling situation? It's a computer. I can consider lots of things and filter them down. What tools are not good at sampling? It's a human. Our ability, I said these numbers early on are beyond what we can even comprehend. If I asked to, even later on, if I asked a chemist to write down 10,000 molecules, it's not going to happen. And so, given that this is a sampling problem, and humans, I would say, intrinsically are not great at sampling, I think they're interesting questions to ask, and I'll kind of return to this topic in a bit. Then, if we think about, what are the historical skills of those that kind of work in drug discovery and in medicinal chemistry? So your kind of historical medicinal chemist, almost always, and still would be, trained in synthetic organic chemistry. This is a key skill, molecules need to be made to then be tested. And so having skills that mean that you can make those molecules are absolutely vital for the role of a medicinal chemistry, the role of a medicinal chemist. And I'll come on to, how this historical picture, maybe this is changing over time. So typically smart synthetic organic chemists are hired into a pharmaceutical company, and then they're expected to learn data analysis, compound design, and a general understanding of drug discovery. But being a good synthetic organic chemist is no guarantee of them being a good medicinal chemist. To me, as I got on this slide here, that the best medicinal chemists are often those who are expert in design, where the synthetic aspect is only one component of the things they're thinking about when designing a molecule. And actually many medicinal chemists don't really pick up, they pick up limited or no computational skills, when there are almost certainly methods out there that could help them be better medicinal chemists. Yet there's often a gap here. If we then flip that and think about the skills of a computational chemist, so the primary skill set of a computational chemist has also been a technical one. it's been that of computer science. And they, you know, many computational chemists may have done a chemistry degree, but they may have swapped out synthetic organic chemistry options to do more in physical or computational chemistry. And there's historical reasons for this, that computational tools either didn't exist or they were difficult to use. And so you needed those technical skills to be able to do the job. So, you know, if you go, you know, keep going back, if you wanted to do simple, you know, what we might consider simple things today, like, you know, like a substructure search or something, but you didn't have code to do a substructure search, you would have to make that. And so historically, you know, these were the skills of those that kind of came in, you know, often the skills that came into the discipline. A lot of what I'm talking about are kind of sweeping generalizations. So it's not meant to be targeting anyone in particular, but just some of the historical biases that have led to where we are today. And so, like the medicinal chemists, you know, you would hire those who are good with computational tools, and then expect them to pick up the skills needed to operate in drug discovery. I think I've heard many criticisms and questioning of the effectiveness of computational chemistry within drug discovery, within a medicinal chemistry context, and I still think there's a lot of work to be done there, and I'll touch upon that in a second. The other thing is that those that come in with a more of a computational background often don't have those synthetic organic chemistry skills, and generally my experience has been that most computational chemists that don't have those skills, often they won't pick up those skills. That, you know, synthetic organic chemistry is difficult to pick up. And when you do it and when you kind of almost live it through working in the lab and, you know, from, you know, an undergraduate, PhD, a postdoc, those become very ingrained in you. And then often it's easier for, my experience has been that often chemists pick up computational tools easier than those with computational skills pick up chemistry knowledge. Obviously not impossible. So here is another general picture of what I think, and based on both personal experience and experience of speaking to others, of how many departments exist around the world where we have, overall, we're all working within medicinal chemistry. And this plot I've got here is a kind of spectrum from pure computer science skills through to pure synthetic organic chemistry skills. And in the middle is the kind of interface between the two disciplines. So as, you know, say a computer scientist picks up knowledge of medicinal chemistry and how drug discovery works, you know, this is then more a computational chemistry CAD type scientist. And the medicinal chemists obviously are not just synthetic organic chemists. They pick up knowledge of drug discovery, data analysis, but only make, you know, some road, you know, steps along this journey. I've got here a couple of kind of radar plots. Back in the day, I used to play things like Championship Manager or Football Manager, and used to get plots like this. It would be like attacking, defending, speed, those kinds of skills. I think it's a nice depiction of thinking about, you know, what is the skill set of any, you know, group of individuals, and where are their strengths. And so the computational chemist, if they've, you know, come mainly with a computer science background, they've, you know, generally most computational chemists don't have a strength in synthesis. But what we're interested in drug discovery is the ability to design molecules and the ability to analyze the data to then help us inform that design. And so our computational chemists definitely have good skills in design and data analysis, but not as good as they could be. And if we think about generally medicinal chemists, they have some data analysis skills. They're obviously strong in synthesis, often weak in computer science, and have a set of design skills too. But the design skills that I'm talking about with the medicinal chemists, actually they're quite different from the design skills of the computational chemists. The computational chemist is able to leverage, you know, enumeration and thinking about the larger picture of the size of the chemical space. And the medicinal chemists, you know, will leverage their experience, understanding of synthesis, focus on the project needs. So actually, if you think about all the skills you might want, they're probably similar, but come from different perspectives. And so thinking about the pure skills, the computational skills, the synthetic organic chemistry skills, actually the value of those are decreasing, I would say, over time. So it's like, calling it the democratization of computational tools, that over time, these things have become easier to use. So me personally, I'm a computational chemist, but I don't code anything. I don't use Python. I do all of my computation in Nine. So there are tools such as Nine. This isn't, a promotion for Nine necessarily, but you can use tools like Nine, which are open source, that enable computation to be done and automation to be done without knowing the language of coding. So it's about thinking like a computer scientist, not knowing the technical pieces of how to be a computer scientist. And so workflows in Nine look a bit like this, where data comes in and the data is flowing through a workflow, and each of these nodes does a certain task. In this case, it's doing an R group decomposition where we define a scaffold of interest, and then we can say, well, give me R1, give me R2, give me R3, R4, and then output that as a table. So you're no longer is there the need, you need to know what you want to achieve, but you don't need the knowledge of how to actually code it up, making this much more available to people who don't have a computational background. The same can be said for synthetic organic chemistry. So it used to be that basically all synthesis was done in-house, and so synthetic organic chemists were hired to make molecules in the pharmaceutical companies in their labs. But today, actually, there are many options for external synthesis, many CROs who can provide great services, or there are options to use automation. So I used to work at Lilly, and here's one of the papers from the automated synthesis lab at Lilly, and Lilly have made significant investments there, where you can use robotics to do the manual aspects of synthetic organic chemistry. Or you'll see publications from Lee Cronin of similar things, of setups where ultimately you may be able to get to a situation where you give a molecule to these systems and they come back having made them via robotics. Another thing is that the knowledge of synthetic organic chemistry is being more kind of codified. So prediction of synthetic routes, this is getting to the point where it's useful, and actually it will only improve. And an analogy here is, the knowledge of routes in London of black cab drivers, that with the arrival of a sat-nav, the value of knowing the route from point A to point B, the value is less. It's not that there's no value, it's that there are other options which are now enable other people to come into the field, and where we see the likes of Uber appearing in big cities. And actually, to me, this is a direction where things are going, and it will only increase, where more people will be able to access molecules than just those that have expert synthetic organic chemistry skills. And so that picture I showed before, where we had the peaks around computational chemistry, peaks around medicinal chemistry, to me, these groups will come together eventually, where our medicinal chemists, more of them will have knowledge of computational methods, have those methods available to them to use. And our computational chemists will have a much better understanding of how drug discovery works and all the pieces and how to drive those projects forward. And so those skill sets that I showed before that were, okay in data analysis and okay in the design, but with strengths in synthesis or computer science, actually those strengths will move towards data analysis and design with less of an emphasis on the pure skills of computer science and synthesis. And having more people in these spaces, I think, will then enable higher quality molecules to be designed, which should push projects forward more quickly. And so to move to a more mixed skill set model, at the moment, I think medicinal chemists could make use of many computational tools, but often they don't. So you've got a largely kind of naive group, and the uptake of existing tools is just as much of a challenge as the development of new tools. So anyone who is working in developing new tools, thinking about how that's going to be, actually used, I think is a key piece. Not just develop a tool, publish it, and that's job done, that's really the job's just beginning to then allow that to be leveraged in a practical environment. And so key pieces to achieve this I see as being communication between those two groups, a lot of it being education. As a computational chemist, I spend a lot of time teaching medicinal chemists to use computational tools such as KNIME, but also structure-based design and other pieces too. Support from management is key, where there's clear direction that the department will move in this direction, again with a vision. So that this is a vision, it's my personal vision. Others may have different visions, but I've seen management that has a similar vision to the one I'm describing here. And then over time, you kind of achieve a cultural shift. If you get success of members of a team from using a computational tool, suddenly other people get interested in how they might have achieved that. And so over time, you can achieve, entire departments operating in a different way to how they'd done before. Thinking then about the skill sets, that actually being in medicinal chemistry, so if you want to operate in medicinal chemistry, I see the pure skills of either synthetic organic chemistry or computer science, that over time those will reduce in value, and that they're being either commoditized or democratized. And that it will become those who have those mixed skill sets that have the highest value, where design is the ultimate objective, and it comes from a combination of these pieces. If you're an experimentalist, I'd always recommend you to go and gain some data science skills. I've seen plenty of examples of that and the impacts of doing that. But finally, let's not forget the human skills that have and will always be hugely valuable. Whether it's empathy, leadership, communication, kindness, all of those things to be overall impactful within a team environment. If we think about the roles of the skills in more detail of human scientists, and then automation approaches, so either robots for doing things or computers for thinking about things. These skills are really complementary. So if we think about the strengths of a human, it's really that ability to see the big picture, to take a step back, think about strategy. The flexibility that a human brings, with humans with a pair of hands can do a huge number of different tasks. They're able to bring in information from elsewhere and be selective on that. So, chemists going, oh yeah, we had found this change on this other project, I remember. We also changed it to this. That insight is useful. The other thing is then operating within uncertainty. You sometimes just have to take a punt. You just have to trust your gut and go, let's do this because it feels right. Computers can struggle in those spaces. They want certainty. The strengths of a robot or a computer are really the opposite of the weaknesses of a human. So computers or robots are really good at handling large data sets or giving them a huge queue of things to do. They don't make mistakes. They can work 24 hours a day. Another thing they're good at is optimizing against multiple endpoints at the same time and balancing and making trade-offs between those things, and doing that without bias. That they will do what they're told. And so the weaknesses of these are really the strengths of the other one. That the human struggles with large data sets. They struggle when they're given a million things to do, and get tired after a period of time, can only work for 7 or 8 hours a day, struggle to consider multiple things at once, have intrinsic bias. Whereas the computer is geared up to do the thing, or the robot is geared up to do the thing it's been configured for, and it'll only do what it's been told with the inputs that it's been given. So if there's information that could be useful to it, unless it's actually programmed into what it's going to do, it won't take advantage of that. And often you find, especially in medicinal chemistry, you create a large library of compounds, and then it will prioritize a number of things with comparable scores, and will struggle to differentiate on them, when actually the best idea might be 50th in a list of 100, but it might have considered 100,000 coming into it. And so a human can have, a human and a computer together can be highly impactful. There's a quote here, so that computers are good at tactics. That's actually, what is it exactly you're going to do within a set of constraints. and that humans are good at strategy. So the idea is going forwards that humans decide at a high level what we're going to do, but then to optimize each individual piece of that puzzle, the computer then can hone in on tactics. And so some of this is this idea of redefining medicinal chemistry. So historically, humans have led this process where it's typically look at the SAR, where SAR is structure-activity relationships, really look at the data, and then just think about, okay, what are we going to make next? And there's only, as I've said, that's a limited pool of things to consider, and based on the experiences of that individual. So this can be redefined from that narrow space to think, okay, well, let's enumerate a huge space, and then let's predict and then prioritize. So the computer can give a prioritized list of compounds, and then a human may be able to inspect and say, oh yeah, that's not something I'd thought about, but look, it looks like it should tick the boxes we're interested in. And so what we want to achieve is really a synergy between the human and the computer, accepting that actually the generation of ideas doesn't suit the humans. There are, medicinal chemists who are good at design, but it's not an intrinsic strength of a human scientist. And that by the computer can enumerate big spaces, and then filter it down, as I've said. And the reality is actually, this historical mode, there's still plenty of opportunity for that, but often this more enumerate and predict often doesn't happen within medicinal chemistry organizations. And I think there's an opportunity for that to play a role in the way we do medicinal chemistry. In this mode of enumeration, this is where they're thinking, I've got to call this slide thinking like a computer scientist, where often we'll use reductionist type approaches to try and simplify the problem. And so the challenge with enumerating chemical space is that you get into these issues of combinatorial explosion. So imagine this scaffold here, it's got 15 heavy atoms. And so we could imagine growing this quite significantly at multiple different vectors. If we've got 10 available positions, and say we imagine 5,000 fragments at each one, if we did every possible combination of all the fragments at every position, we get 5,000 to the power of 10 possible molecules, which obviously is a huge number. If we do things like start to constrain this, let's limit to just 2 positions at the time. We have 5,000 to the power of 2, and then 2 to the power of 10. So we're at about 2.5 billion molecules, which is still quite a large number and difficult for some methods to handle, but on the order of where we might be able to handle that with current computing power. Another option to kind of navigate this space is think about sampling. You'll see methods in the literature for sampling this large chemical space. But you do run the risk of missing things whenever you do a sampling type approach. Another option is then to constrain this search further to try and limit the numbers. And this is this kind of reductionist type approach that I'll talk about on the next slide. So this is where we think about using this matched molecular pair concept. So medicinal chemists often operate in matched molecular pairs. And a matched molecular pair is a single point change. And actually Al's speaking next, and we'll almost certainly hear about matched molecular pairs, where we make a single change at a time. And then by making that single change, we're able to rationalize the difference in observations that we make. We can then attribute those changes to the change that we've got. One assumption here is this assumes additivity of our SAR, the changes that we make, that if we make A to B at one position and then B to C somewhere else, if we do A to B and B to C, that those will be additive. But often that assumption is valid, and operating in a world of non-additive SAR can be highly challenging. And so what I've done is the idea is that we create enumerations to create every possible match pair from a given starting point. We do lots of different perturbations to those molecules where we might change atom types, we might form rings, expand, contract rings, as well as add fragments at available positions. And so the idea is that, if you had a chemist looking at a scaffold thinking, oh, what can I do with this? Is that you try and create a set where everything they could possibly imagine is enumerated in that set. And giving an idea of space, if you give a single molecule to these sorts of approaches, you would typically blow up sort of 1 to 200,000 compounds, which is a number that's perfectly manageable by a computer. And then the idea is you might filter this set, you know, if you know some of the SAR, we can't put a substituent there. We have to maintain this acceptor at this position. You can put that in and filter these further, and then apply models to this set and prioritize a set of compounds. Finally, so this kind of moving to be, you know, this mix of medchem and compchem, I think can happen today. That you know, I think things are moving in that direction slowly, but there's more examples of you know, computationally savvy medicinal chemists and more medicinal chemistry savvy computational chemists. Next, think about, well, you know, how do computational tools interact with users? And so I see many examples of people planning, you know, slick user interfaces, training people to use it, trying to get the layouts just right, trying to go, okay, you know, how can we get people to run good, you know, valuable calculations with the minimum number of clicks? And actually, sometimes the issue is just getting people to know that they should be running this approach at all, given the data that they have and that it could be useful to. And so what if you had a system that required no clicks to get something done? You know, what could that look like, where without asking, the computational method knows it should be run, it happens, and the results are sent to you. So this is around, you know, around automation. So here's a vision of mine around automation. It's that every time any relevant new data appears for any project, that data is summarized, that the analysis is done, and then it's sent to the people that need to see it in a format that's easily digested, and that happens almost instantly. So people are getting input of things that are going to be useful to them without asking. The assay submissions are made when predefined criteria are met. So, you know, if new data's come out, this compound's potent enough, it just goes straight into the next assay. There's no human catching that and then sending it off, which can cause delay and it can cause mistakes. And the computational methods, that they're automatically triggered. And this just happens. And that all of this happens in the absence of anyone asking for that, allowing human scientists to get on with creative tasks and thinking about strategy, like we spoke about. And so I published this paper along with my colleague Matt in 2019, and it's around applying a kind of virtual assistant type concept. So the likes of Alexa or Google Assistant, or, you know, many tech companies have these kinds of virtual agents. And the handle turning basically is carried out by a robot or a computer. You know, the computer carries out the thought tasks, that they're able to identify that new data has appeared. So if new data appears that's relevant to a project, the agent is able to identify that new data, analyze it, send it to the team, but then continue and go, okay, well, given this new data, what compounds will I design? Let's automate the compound design. Let's assess them. Let's run those computational models, send the results to the team. We can predict retrosynthesis. So we have a system where we can identify new data, design molecules, propose them for synthesis, and then if you have an automated synthesis platform, send them off for synthesis. And that this cycle, this is mostly tactics, right? You've set an objective and you want to iterate over it to meet the objective. This can be fully automated, and in 2018 we achieved a kind of fully automated cycle of this design, make, test, analyze, DMTA medicinal chemistry cycle, you know, had achieved, you know, been fully achieved without any input from any intellectual input from a human scientist. I'll skip this, but just mention what it is. Here's the example of how we were interacting with users. But the main message here is interact with people where they're looking anyway. So every scientist within a drug discovery organization has Outlook or another email inbox on their desktop to interact with them in those spaces. So you don't need a new user interface. You can just ping a message to someone with the detail that they need. This email is a little bit crude, and it's a screen grab from the paper here. It's just a proof of concept. Like, useful information can be sent out, and then you can have links to tables, to graphs, to whatever that might be, and mixing computational approaches with just data analysis. And finally, could a fully automated future exist? So where currently humans act as gatekeepers between assays, and that if we had a process that could continue in the absence of human input, but where humans can intervene at any point if they want to, that I think is an interesting future where where you have a system that is both automated but can leverage human input when it makes sense. I think the technology exists to do this, but in the future, could this become the norm? And what skill sets of people would be needed in this different environment? So the takeaway message is really that apply machines to what they're good at, and that's things that revolve around tactics. So robots carrying out repetitive manual tasks, computers carrying out repetitive thought tasks. And so we can then free up human scientists to spend time doing the things they're good at and interested in too, which is planning the strategy, the high-level decisions, and making those decisions in uncertainty, which there is a lot of within medicinal chemistry. And that it's the combination of the computer and the human scientist that ultimately is the most impactful. So in summary, drug discovery is complex, but you can break it down into multiple pieces within that process. Currently, processes are very human-driven, but they can be enhanced using a computer. And a more optimal future is one that where these two things, computation and human thought processes, are operating together. And that then leads to think about, what are the skill sets that particularly younger scientists might like to think about and that they'd like to gain that's going to make them most valuable and most impactful in the future. So with that, I'm done, and thank you very much for allowing me to whittle on about my imagination of what a future might look like.